Organogenesis is the third and final stage of embryonic development. Thus far, we have given you a gut tract, but the problem is there's no orientation to your body, other than the direction of your mouth and anus. Organogenesis will allow us to give you a dorsal and ventral side, or a back and front, to your body orientation. This occurs by the placement of your central nervous system. The central nervous system, composed of your brain and spinal cord, are formed in embryonic development through the notochord and the neural tube. The notochord placement will form this axis of the body that I referred to, giving you a dorsal side. For you, your central nervous system is located primarily on your dorsal or back. However, in other animals, the notochord will actually place on the ventral side, meaning that their central nervous system would be located on their ventral side. We'll talk in animal evolution the benefits of that. This folding in that occurs forms the neural tube and the neural plate. These neural plates will actually guide the direction of the formation of the notochord for us. In the cross-section of our frog here, we see the familiar endo, meso, and ectoderms, and then associated with what will become in you and the frog, the dorsal side of the individual, the neural folds and neural plate are forming. Now this folds in and actually produces your nervous system on the inside with a layer of ectoderm or skin on the outside. Of course, this means that your central nervous system is going to be protected by skin, but our neural tube is also going to help us place the spinal cord and the vertebrae for you that protect your spinal cord. How does this form? Well, this forms by something known as somites. Somites form alongside of our notochord so that we can attach muscles, bone, and connective tissues in place so that the spinal cord that we have developed is protected. Now, this is going to form again through something known as a coelom. In a previous session, we saw seal, which we said meant cavity. So a coelom is just a cavity. Here you see in a developing embryo the placement of the somites. The area has enlarged along the dorsal side to make room for the neural tube and the vertebrae that we're going to have to produce to protect it. In particular, the formation of the spinal cord, as we mentioned, and the spinal vertebrae. But the somites here you see with the orientation of an eye here and a tail bud here, these somites are very long and skinny, and they have pairs. If you could see the other side, these are paired up. So what do you think that these are going to become? Here's a chick embryo, very early in development. So a chicken embryo. And you see the somites, this is from the dorsal side, so looking down on the back. If you were laying on your stomach and someone was looking at your back. These somites come off in pairs, as you see. What do you think they're going to become? those somites we'll come back to and look at in a little bit, but those pairings make us think of potentially ribs. We have to keep in mind organogenesis, or the generation of new organs, is extremely regulated. And if it was not, your organs would not be in the proper location. So here are the steps associated with the proper formation of early organs in you, in particular the formation of this neural tube to eventually form your brain and spinal cord. So we saw a similar diagram to this a minute ago. The endoderm, mesoderm, and ectoderm that we've talked about previous, and then this neural tube that forms in the middle. The new thing here I want to draw your attention to are these things known as neural crest cells. This is going to be an important group of cells for the formation of your skull and facial features. And what we see here in our boxer puppy is that those cells, neural crest cells, did not migrate properly. So instead of having a properly formed muzzle in the front, we get a formation of just half the muscle. So in humans, we refer to this as cleft lip. So the connection between the mouth and nose failed to form properly because the neural crest cells have migrated properly on his left side, but on the right side did not. So in our puppy that we saw, the neural crest cells on one side would have migrated properly, and the ones on the other side did not. So any features associated with those particular crest cells that did not move properly wouldn't form the proper structures.